Hello, so I'm a graduate student at Penn State College of Medicine, and I would like to thank you for viewing my poster video. So my poster, titled The Role of Cellular Compartments and Viral Capsid in Hepatitis B Virus Entry, focuses on understanding the early steps of HBV entry. So we know many details about the replication events that follow cccDNA formation, but we know very little about the intracellular viral trafficking steps that precede cccDNA production. So to start off, HBV binds to its cell surface receptor NTCP. It's then endocytosed. HBV traffics to the early endosome and the late endosome, and then it's believed that for productive infection, HBV must escape the late endosome and be released into the cytosol. Then the capsid must traffic to the nucleus to deliver the rcDNA, where the rcDNA is converted to cccDNA. Transcription and translation occur, followed by capsid assembly and reverse transcription, and then either virion release or intracellular amplification follows, which is where the capsid is trafficked back to the nucleus. So of noting, intracellular amplification, this bypasses all of the steps required for infection, shown in this red bracket. So our lab has identified a number of HBV capsid mutants noted in figure 1b that have this unique phenotype in that they fail to form cccDNA during infection, but they can form cccDNA from viral DNA produced into the cytoplasm, which bypasses the viral entry steps, including the endosomal trafficking during infection. So the focus of this poster is on the mutant L60V present at the dimer interface shown in figure 1a. So these results have led us to this overall hypothesis: these mutant viruses have a specific defect in one or more of the viral trafficking steps during infection, possibly within the endosomal compartments. So when testing one of the naturally occurring variants L60V, figure 2 demonstrates how this capsid mutant is not infectious, as cccDNA formation determined via southern blotting shows that wild-type HBV produces a high amount of cccDNA, while L60V HBV produces very little detectable cccDNA. However, upon transfection, which is figure 2b, there is a high amount of cccDNA in both wild-type and L60V HBV, indicating the intracellular amplification pathway is intact. So in order to further assess the role of the endosomal and lysosomal compartments, we've utilized drugs and treatment conditions shown in figure 3a. So we're infecting HBV2 NTCP cells with wild-type HBV, and then treating with drugs that inhibit the lysosomal acidification, such as chloroquine or baflomycin A1. And we found that cccDNA is not affected and may even be slightly increased, as shown in figure 3b. So this indicates that the HBV entry process is pH independent. We then tested with a drug that specifically inhibits the early to late endosomal maturation called EGA. And as we show in figure 4a, we demonstrate that when cells are infected with HBV and then treated with EGA, there's a decrease in cccDNA production. So this indicates that the endosomal compartments are important for entry and also necessary for productive infection. So interestingly, when we infected cells with L60V HBV and then also treated with our lysosomal inhibitors, either chloroquine or baflomycin A1, we found that these drugs do not enhance L60V cccDNA. But they do enhance this novel DNA species detected in L60V, shown by the black arrow in figure 5. So we then further tested the role of the proteasome with L60V HBV entry by treating the cells infected with L60V with proteasomal inhibitors, lactocysteine or MG132. And we found that these proteasomal inhibitors strikingly increase this novel DNA species detected from L60V infection, marked by the black arrow in figure 6. So utilizing a synchronized infection, as shown in figure 7a, followed by subcellular fractionation, we can identify entry kinetics through tracking of the rcDNA via southern blotting. So we can demonstrate that at 24 hours post-entry, nuclear signal is dependent upon NTCP expression. So we plan to use this experimental flow to test the entry kinetics of L60V in comparison to wild type. So these results have led us to our current conclusion model, where wild-type HBV must traffic through the early endosome and the late endosome in order to lead to productive infection, which is cccDNA production. However, L60V is likely misdirected to either the lysosome and or even the proteasome, causing it to never reach the nucleus for productive infection. And with that, I would like to thank the WHO lab and the Wang lab, and then our funding sources for the data that was presented today. Thanks for watching.